Hi, hello. My name is Wouter Peelaerts, and I'm an assistant professor at the KU Leuven in Belgium. And I'm excited here to speak at the Defeat MSA virtual conference. So for this talk, I'll be focusing on a central research hypothesis that is actively investigated in the field, which is the Prion-like hypothesis in MSA research. And so how this is providing a framework through which novel diagnostic tools and novel therapies are being developed. And so many of these developments focus on the role of the protein alpha-synuclein and how this protein is considered to play a central and important role in the disease process. There we go. And so, I'll be talking about the prion concept or the prion hypothesis. And so for that hypothesis, I'll be addressing two other concepts that fit within that hypothesis, which are the synuclein transmission hypothesis and the idea behind synuclein strains. And so I'll be giving examples from experiments that we do in the lab, from experimental models, and what this means for disease, for both the transmission hypothesis as well as for synuclein strains. And so maybe just as a reminder, I probably don't need to explain what multiple system atrophy here is, but in MSA, we know that there's this really important key pathological feature, which is that alpha-synuclein, the protein alpha-synuclein, is aggregating and accumulating in oligodendrocytes. And so these are very specific and peculiar cellular lesions that you find in the brain of a person with multiple system atrophy. And so this is very unique and very specific for anyone that has MSA. So here in this slide, you can see the brain or the striatum from a normal control subject where we looked for deposits or aggregates of alpha-synuclein. And you can see that on the left side, we don't find any alpha-synuclein, or we don't see any of that protein. On the right side, you can see a lot of those inclusions. You can see white matter regions where a lot of these cells have aggregates of alpha-synuclein, and these aggregates are found in a very specific cell type, the oligodendrocytes, which are the white matter producing cells of the brain. And so this is key. This is very important for multiple system atrophy. And so we've been studying this protein alpha-synuclein for a very long time. So again, we think it's crucial to the disease. And so synuclein is in fact a very small protein that is expressed in our brain. It's expressed throughout the body. It's expressed in immune cells. You can also find it in red blood cells. So you can literally see it throughout the entire body. And so it's a small protein shown here. So you have these synuclein monomers, which are single molecules of synuclein protein. And so if you take a picture of that with an electron microscope here, shown on the bottom here, you can see very small dots. In fact, it's so small, so tiny that you can't even see it. And so this small protein doesn't have a distinctive shape or conformation. And so that allows it to freely, reorganize and freely bind with other proteins, including itself. And so once it does that, it forms these what are called oligomers, so small molecules or aggregates of synuclein proteins that are packed together. And so once these small oligomers, these small assemblies, small aggregates of alpha-synuclein keep existing, and when they keep binding with these synuclein monomers here that you see there, they can also form these larger filaments shown here on the right. And so these are, again, very specific for synucleinopathies. And so once these filaments start building up inside a cell, for instance, they start really packing up and start accumulating within a cell body. And this is when they form these typical inclusions that you can, for instance, see here on this slide that we can see with a conventional light microscope. And so what is really specific and important for these filaments, something that we've discovered over the last 10 years of doing research here, is that these filaments, these fibrils of alpha-synuclein, that they can replicate. So they can break in smaller pieces, forming these smaller, what we call fibrillar seeds or nuclei that can seed again with monomers that are present in, for instance, a cell, and they can form new filaments or new fibrils of alpha-synuclein. And so it's almost like these proteins suddenly have become infectious and can amplify and replicate within a certain environment and basically sustain this amplification and replication process just on their own. So what this means in a cellular context is that we believe that in response to a certain insult, an infection, inflammation, pesticides, pollutants, that cells can actually react and respond to this by expressing alpha-synuclein. So you have a certain trigger that causes synuclein to aggregate and accumulate within a cell that is exposed to a certain trigger. And so these cells will then start expressing alpha-synuclein. They will start expressing the protein. And these synuclein proteins will then start to aggregate and accumulate within the cell to form these small assemblies, these small aggregates that we know that can keep replicating and amplifying. And so these aggregates can then literally escape a cell and then transmit to a neighboring cell. So they can transmit from one cell to the next, where then the same process can happen over again. So these neighboring cells, they will take up small aggregates of alpha-synuclein. These aggregates will amplify within the second cell, and more of these aggregates will form, and more of these aggregates will then amplify in the cells that are connected with each other. And again, this can continue until these large inclusions are forming, until cells are actually developing so much and forming so much of these aggregates that they start depositing within a certain cell. And this, again, is when we see these inclusions, when these typical inclusions form, for instance, in oligodendrocytes in MSA, or in neurons and in Parkinson's disease or other synucleinopathies. And so during this process and along this pathway of synuclein transmission and propagation, cells will actually suffer, because the process of forming these aggregates, we now know, is actually toxic. It's harmful to cells, and especially when this amplification process or disaggregation process happens in cells that are specifically vulnerable, these cells will then also start dying, like oligodendrocytes in MSA, or again, specific cellular populations, for instance, like dopaminergic cells, which are very, very vulnerable cells in Parkinson's disease that then eventually will degenerate and die off. So this is a process that we study in the lab, and the way how we study this is by synthetically making these synuclein monomers in test tubes. So we make these in the lab, and so we synthesize these monomers, for instance, here, as you can see them. And what we do is we put these monomers in a thermomixer, not the thermomixer that you would have maybe at home in your kitchen, but the one that we have here in the lab. And we would shake these monomers for several days, here, shown here, in the lab, and then make these larger aggregates of alpha-synuclein. So by shaking them over time, these actually small single molecules will then start binding with each other. They will form these oligomers, and then over time, these smaller oligomers will turn into larger fibrils and large filaments. And when they come out of the reaction, and if we spot them on a grid and take a picture with the electron microscope, you can see here these fibrils deposited here on the grid where we spot them, and how we take these pictures. And then for our experiments, we sonicate these fibrils and turn them into smaller pieces, what we call preformed fibrils. And then they are ready for doing experiments. And so the way how we do our experiments here is, for instance, by injecting what we call these alpha-synuclein preformed fibrils, by injecting them in an animal or a mouse brain. Very often we inject them in a region called the striatum. So this is actually shown here along this dashed line here, where a needle just goes in, and where we inject these aggregates, these assemblies of alpha-synuclein in the brain of these animals. And so if you turn the brain again 90 degrees this way, if you look at a coronal section, for instance, like this is where we would, for instance, inject these synuclein preformed fibrils. If you then wait for a couple of months, what you then see is that these animals start developing typical pathology and those typical inclusions that we would see in a patient brain. So a mouse injected with fibrils, here on the right, you can see cells with these deposits with these aggregates. A mouse that was not injected with these fibrils does not show any of these typical inclusions or these pathological lesions. And so more recently, we've done an experiment where we injected preformed fibrils in mice that were genetically altered. And so by injecting these in mice that were expressing synuclein monomers in oligodendrocytes, we asked whether these fibrils might or might not be important for multiple system atrophy. And so here you can see a picture of mice that were injected again here in the striatum. So that red dot is the injection site. We have mice that were injected with a control protein, and we have animals that were injected with synuclein fibrils. Here in the control animals, you see the striatum, part of the brain that is heavily affected in multiple system atrophy. But here in these control animals, you can see all of these white dots, the white matter that looks fine, that looks normal. You can also see the corpus callosum, this white matter rich region, the ventricles, and so forth. The animals that were injected with the fibrils, this is only 9 months after these mice were injected, do not have any of this white matter anymore. So there's a loss of oligodendrocytes, there's loss of white matter, the corpus callosum has almost vanished, and the ventricles are now enlarged, and the striatum is atrophied. And so this all comes with the formation of these aggregates, these inclusions in oligodendrocytes, and these mice also have significant motor deficits that reminds us of multiple system atrophy. So by simply injecting these synthetic synuclein fibrils in the brain of these animals, we can actually mimic MSA experimentally in the lab. So suggesting that there is an important role of these synuclein aggregates in pathogenesis or the disease process of MSA. So, so this is more of a conceptual idea, an experimental idea that we've been testing. And so what does that mean in let's say, in a more broader perspective, more clinical perspective? And so another idea that we have is that synuclein transmission, or that the Prion hypothesis might also explain why there is such heterogeneity between synucleinopathies, and that it might also explain where these diseases might start and how they progress and how they develop over time. So the idea that we have is that in response to environmental pathogens, which again can be bacteria, which can be viruses, which can be pesticides or pollutants, that cells that are directly exposed to these pathogens will start expressing and accumulating alpha-synuclein. And so pathogens can enter the body through various ways, through the nose, through the gastrointestinal tract, or in case of multiple system atrophy, we're thinking about the urogenital system. And so once that happens, synuclein would start to aggregate and accumulate, for instance, in the urogenital tract or in the gut or in the nose. And in certain conditions, in certain predisposed individuals, these aggregates that would accumulate within these sites are then prone to escape from the periphery and travel to the central nervous system, where they then can instigate or trigger disease. And so there are several groups that have tested this idea, and I'm going to give examples for all of these 3 different routes through which the disease might start and might travel, and through which it might cause this disease, to illustrate this idea behind the Prion-like hypothesis. So first, I'm going to start with the olfactory bulb, so the olfactory tract. So so this is a brain structure that sits very close to the olfactory epithelium. And so when you look in the olfactory bulb, you can already see that there's quite some alpha-synuclein and some smaller aggregates that are present in animals, in humans. So these are actually control subjects that didn't have any disease or any type of dementia. And you can see that there's already quite some alpha-synuclein present here in this specific area in the brain that is very close to the outside environment. And so one question here that was addressed by Nolwenn Ray, who was in the lab of Patrick Brundin. So Nolwenn spent several years of her postdoc studying if synuclein could actually spread from the nose to deep brain structures. And so whether it would mimic, the different stages that one might see, for instance, in this case, for Parkinson's disease. And so what she found is that indeed, if you inject synuclein fibrils here in the nose, again, the olfactory structure here in the top here indicated by that red dot, that if you only inject a single bolus of alpha-synuclein fibrils within the olfactory bulb, that 1 month, 3 months, 6 months, or 12 months after injection, pathology would develop and spread from the olfactory bulb to deep brain structures. So 1 month after injection, you can see that the olfactory bulb develops inclusions of alpha-synuclein, and also connected areas you can see have some pathology there. 3 months after injection, you can see that those same areas are still affected, but now the pathology is also spreading to the other side of the brain. At 6 months, you can see more deep structures being involved, such as the hypothalamus or the thalamus, and again, more pathology in those initial structures. And then even after 12 months, you can see that the pathology has spread all the way up to the midbrain, even affecting regions and structures in the substantia nigra, the locus coeruleus, or the Raffin nucleus. So showing that indeed the disease can spread from a defined anatomical structure to very deep brain structures, and actually mimicking the way how the disease would develop in humans. So another way how we think that the prion hypothesis or the transmission hypothesis might explain how the disease might start and spread and develop is by looking at the gut and by looking at transmission from the gut to the brain. And so a lot of research also has been done in this context. This is work from Vivian Labry's group, where they looked at synuclein aggregates in the gut of normal people and people with Parkinson's disease. And so all of the red color here, the red staining, is a stain that was done to show where alpha-synuclein is. So actually this is a piece of the appendix, where here you can see that there's lots of synuclein in the enteric nervous system. There's lots of synuclein in the vasculature. There's even synuclein in the gut mucosa, showing that synuclein is also expressed in cells, neurons there as well, but also in immune cells. And so what the lab of Ted and Melina Dawson then investigated is whether fibrils could spread from the gut to the brain. And so what they did for that is they injected synuclein fibrils in the gut of mice and examined whether these mice would develop Parkinsonian symptoms and whether they would develop inclusions in the brain after time. And so what they found is, again, indeed, that this was possible. And so by injecting fibrils here in the duodenum, they were able to show that already shortly after injection, pathology would be observed in the brain. Another important experiment that they did is they also cut the vagal nerve, which is a nerve that connects the gut to the brain. And by sectioning, or by performing vagotomy, they were able to prevent the spread from the gut to the brain. So showing that these aggregates of alpha-synuclein travel through neuronal connections from the periphery to the brain. So if these connections are not present, the disease doesn't spread to the brain anymore. And also importantly, what they showed is that when they injected fibrils in mice that were, again, genetically altered to not express any alpha-synuclein, so these mice do not have any alpha-synuclein present, that again, the pathology is not present anymore. So if they inject the fibrils into the gut, the disease doesn't spread anymore. So showing that it relies on this infectious mechanism where fibrils will seed and aggregate and amplify with the available synuclein monomers that are present in neurons and in the brain. So again, providing support that that pathology might be initiated in the periphery and then travel to the central nervous system. And then here, for multiple system atrophy, this is something that we've been examining for quite some years now, is how synuclein pathology might travel from the urogenital system to the brain. And so the idea here that we have is that there is lots of pathology in the urinary bladder, especially synuclein pathology, synuclein aggregates, as well in normal individuals, but also in people with MSA. And that in response, for instance, to urinary tract infections, that synuclein starts to aggregate and accumulate in the urinary bladder. And that again, in certain individuals, these aggregates will then spread from the urinary bladder to the brain. And so there's a lot that we did there. One of the experiments that we did, but also other researchers did here, for instance, this is an example from a Chinese team, is they injected, again, alpha-synuclein fibrils in the bladder, the urinary bladder of mice, and then examined whether these aggregates were able to spread from the urinary bladder to the brain. And indeed, again, what they found was that after injection of these aggregates in the urinary bladder, they found that these aggregates then started to travel and started to accumulate and amplify in regions that are typically affected in multiple system atrophy. So the pons, the midbrain, and different areas, different sites in the spinal cord. So we have also done something very similar where we took and amplified and isolated fibrils from the brain of people with MSA, amplified them in the lab, and then injected them in the urinary bladder of mice. And what we again found was that after injection of these fibrils, these mice then started developing symptoms and features that were reminiscent of those of multiple system atrophy. So again, showing that from the urogenital system, the disease might propagate from the periphery to the central nervous system. So this prion hypothesis, or this transmission hypothesis, actually provides a framework to explain how the disease might start and why these diseases might be so different. For instance, why people might develop Parkinson's disease, Lewy body dementia, or for instance, multiple system atrophy. All right, and so this brings me to a second part of this talk, just a few slides that I still have on synuclein strains. Another important concept in the prion hypothesis. So I'm just going to give just one or two examples from experiments and just talk a bit about why this is important for diagnostic and therapies. And so a question that that really intrigued us again quite some time ago is that in synucleinopathies, you'll see that again, synuclein has this really important role, and that this one protein is involved in so many different diseases. But at the time, what we didn't understand is why you have this one protein, but so many different diseases. If there's one protein involved and if there's one cause, why aren't we seeing a single disease? And so the idea that we had to explain this is that there are different strains, protein strains of alpha-synuclein, with different functionalities and different pathological features. So different types of synuclein aggregates with different conformations that would exist in people with MSA, or it would exist in people with Parkinson's disease or Lewy body dementia. So instead of focusing on these very large aggregates or deposits of alpha-synuclein, we were now focusing on what was actually inside those aggregates and how those fibers were built. And so only very recently it's been possible to actually solve the structure of these fibrils. So this was done by the team of Sjors Scherens and Michel Goddard, where they isolated and purified fibrils from the brain of people with MSA, and they were able to solve the structure of these filaments, of these fibrils of alpha-synuclein. And what they found was actually quite striking. Is that in multiple system atrophy, you will see a type of fibril that is composed out of two big filaments. So here we're actually looking at the fibril from the top. So we're looking top down here. So here, this is actually the side of the fibril. This is the top of the fibril. So we're now looking from the top down here on the fibril itself. And you can see one molecule here that is sitting here on the top, and another molecule of alpha-synuclein here that is sitting on the bottom. And so these are actually two very long rods of alpha-synuclein that are twisting together and are turning around a central axis. And so this is very specific for multiple system atrophy, because if you would take fibrils and isolate them and purify them in the same way from another person with MSA, you would find the exact same structure. And that's really interesting, because if you would do the same thing from a person with Parkinson's disease, and again look at the same fibrils, you will see that these structures are actually different. So instead of two molecules, now you have only a single molecule. So we're looking at a single filament, and so the structure is therefore different. And you see the same thing from a person with Parkinson's disease with dementia, or a person with Lewy body dementia. You can see that same structure, but still very different from the structure that we would see, for instance, in fibrils from people with multiple system atrophy. And so that idea or that concept is really important, because if the conformation or the structure of these, what we call synuclein fibrils or synuclein strains, is different, it will also mean that would come with different functionalities and different pathologies. Because already some time ago, we did a couple of experiments where we showed that if you use different types of synuclein strains, so here called synuclein fibrils, and here another strain called synuclein ribbons. So this actually looks like spaghetti. The other one looks like, flat and twisted tagliatelle. And if you use these fibrils and inject them in an animal's brain, what we found was that these animals would start developing different diseases. So even though this is the same protein, alpha-synuclein, just because of the way how the fibril is built up differently, these animals would develop different diseases. And in fact, in the mice injected with the fibrils, we noticed that these mice started developing more Parkinsonian symptoms, whereas the mice that were injected with these other synuclein fibril, the ribbon here, they would start developing Parkinsonians with MSA-like symptoms. So we know that this is really important, and we know that this can actually drive different phenotypes and different diseases. And so now I'm just going to show a couple of examples of why that is important, and a couple of examples of how this Prion hypothesis has actually provided a platform for people now to design diagnostic tools and develop new therapies. And so here again, you can see an image of a synthetic fibril that was developed in the lab. So this is actually from a study that just came out very recently, where the team here developed a compound or a molecule here called FO5O2B. And they actually selected this molecule or designed this molecule to very specifically bind with this particular type of fibril. And so here you can actually see it if you look very well. You can see it to bind with here, this particular groove here that sits on the outside of the fibril here and here as well. And so by designing the molecule in such a way that it specifically binds, they were also able to, for instance, to add a tracer, or in this case, a PET tracer. And so when they administered this tracer, this molecule, this PET tracer, in this case to a non-human primate, you would see that in normal non-human primates, you would here see the brain, and you would see that the tracer doesn't really specifically bind to anything. But now when the tracer is administered to an animal that was injected with preformed fibrils, you can see that the tracer now starts binding with these fibrils, and you can see signal here throughout the brain of these animals that are developing pathology. So again, this is really important because also here you can see another example. So this is a PET tracer that was developed by AC Immune. This was a PET tracer that, again, that was designed so that it binds with synuclein fibrils. You can see here two control subjects that were injected with the tracer. You can see the cerebellum here, and you can see that these people don't necessarily or don't exactly bind or don't show any accumulation or any binding with the tracer. But then here in the bottom, you see two people with the cerebellar form of multiple system atrophy, so MSAC here on the left and the right. And you can see that here in the cerebellar peduncles and the cerebellar white matter, this tracer is now binding probably with synuclein fibrils. So by designing something that is specific for synuclein fibrils, it has now become possible to actually look where pathology is inside the brain and whether people might or might not have a multiple system atrophy. And then I was going to explain this slide, but I just see I'm running out of time here. So I'm just going to quickly jump to the conclusions here. So I think I was able to illustrate that alpha-synuclein is really at the center of MSA pathogenesis, that this is a protein that can aggregate and then acquire infectious features, that it can travel from cell to cell, and that might explain the progression of disease in different types of synucleinopathies. That this is also linked with neurodegeneration and disease progression, and that this idea of strains and this idea of the Prion hypothesis is now again providing us with something to build on and develop new diagnostic tests and novel therapies that are actually entering the clinic, and that is something that I'm really excited about. And then finally, I'm going to finish here with this slide. Of course, I want to thank a lot of people, especially the families and the patients who were really instrumental in this research, the labs that were involved in this research, and then of course all of the funding agencies that also have made it possible to do all of this research, and of course all of you listening to this presentation. Thank you very much.